Well, thank you for coming today. I am presenting on nitrogenoma eutropha. I wanted to do a study and find out, we actually started as a class project. It was a group effort to begin with, and by the end of the spring semester of 2015, we were actually able to start getting the nitrogenomas to grow. It took almost an entire half of a semester from the time we got the product to be able to get it to grow. I chose to continue on with the project beyond spring semester, and thanks to the effort of several students in our class, I had some wonderful growth to work with throughout the summer and ongoing for this project. We looked at the nitrogenomas to see, number one, what type of bacteria it was, what it metabolized, if it would do what the product we were testing, which was a probiotic skin product, would do what they thought it would do, which is reduce the smell of the sweat and be beneficial to the human skin flora. As I went through, we identified it to be a Nitrogenoma eutropha. It's a gram-negative bacillus, and it is a chemolithograph, which means that it will change pigmentation based on temperature changes, stresses to the bacteria, and it's able to use carbon dioxide as a carbon source and metabolize inorganic substances, in this case, ammonia. It is a highly motile bacteria which utilizes a flagella for motility. We were unable to get actual pictures on the slides of the flagella due to the staining procedures that were required. Its optimal environment is a dark environment of approximately 21 degrees centigrade, and it likes a mildly acidic environment of a pH of 6. When the pH or temperature is less than optimal, it will produce chromophores, will change pigmentation, and will turn into a fluorescent pigment. As I mentioned before, the purposes of this study was to identify the morphology, the bacteria, the metabolism, to produce slides that demonstrate the genus Nitrogenomas and whether it's gram-positive or gram-negative. We also wanted to test the susceptibility of various potentially pathogenic bacteria to the Nitrogenomas and how they interacted, and to check the Nitrogenoma eutropha's susceptibility to various soaps and hygienic products that are used today. We also wanted to interpret these results into an educated conclusion on how there were plentiful Nitrogenoma eutropha cultures, which were used to historically be within our normal skin flora prior to the use of current hygienic products, and how we could promote the recolonization of this bacteria and the benefit that it would be to the human population. We began by isolating the Nitrogenomas. We did this by placing various concentrations of the probiotic skin product that was sent to school using sterile pipettes into a growth solution. We eventually did get growth using 1,000 microliter concentration. And once we isolated that, we worked on identifying the bacteria present and growing. It was identified again as Nitrogenoma eutropha. We then broadened our study in experiments. Once we were able to isolate growth, we studied the pH in which it would have the best growth. We inoculated a pH tubes of 2, 4, 6, 8, and 10. This was done during the spring semester of 2015 as a group. We discovered that the pH 6 broth inoculated with the 1,000 microliter concentration had the best growth. As you can see, the pH 6 tube shows a turbidity and slight pigmentation of the broth. This indicates the growth of the Nitrogenoma eutropha. Once we determined the pH, we looked at the salinity, like whether it would be considered a halophil, liking a saline solution, or more neutral. The growth was placed into tubes with salinities of 0%, 5%, 15%, and 20%. The best growth occurred in the 0 and 5% salinities. This makes it a neutral to a very slightly halolytic bacterium. Once the growth was established in the broth, we went through a very difficult time of trying to get it to grow in a nutrient agar. We eventually were able to do this by using 4 loops, sterile loops, of a sample from a pH 6 broth that showed visible growth of the nitrogenomys. We incubated them at 37 degrees centigrade initially, showed that it had a translucent mucoid appearance of the growth, and the pigmentation varied whether it was incubated at room temperature or at 37 degrees centigrade. If it was incubated at room temperature, we tended to have a more greenish pigmentation. If it was incubated in the incubator at 37 degrees, then it was more of an auburn colored. As you can see by the different plates, we have this was incubated at 37 degrees, so a more auburn pigmentation of the growth. You can notice that it's translucent and mucoid in appearance. Over here, we have it incubated at room temperature, or approximately 21 degrees centigrade, in which we have a more greenish pigmentation. And then on this, you can notice the streak plate, which we have 3 streaks that are done. This is in hopes of isolating a colony. In the very center, you can see isolated colony where the pigmentation is darker in the middle of the colony and lighter on the outside. This also shows that the Nitrogenomys eutropha is a motile organism and will grow outward from the initial inoculation. Once we were able to get the growth going on the nutrient agar and the pH broth, we worked on doing microscopic studies. You can see with the staining that it is a gram-negative. It is a bacterial shape, in other words, a rod shape. This is with the Nitrogenomys more spread out. Over here, you can see the Nitrogenomys have a tendency to clump together, and you can see the various clumps. Over here is just 100 times magnification, and over here we have 100 times magnification plus a 2 times zoom on the camera lens. We were able to have one slide that demonstrated the ability of the Nitrogenomys to produce chromophores. If you can see right here in the circled area, we have bacterial cells that show a slight fluorescence compared to the surrounding cells. This slide was made with a sample that was taken directly from the refrigerator. We tested how the Nitrogenomys would utilize nitrate, and you can see growth in the nitrate test. And in addition, which it might be difficult to see, there is a bubble in the dextrose tube, which demonstrates that it does produce gases. Now, the gases that it produces are non-nitrogenous. But it does produce a gas when it is utilizing the nitrates and breaking it down. We did an intro-tube 2 test to determine the different types of sugars that the Nitrogenomys eutropha might utilize. It was negative for glucose, lysine, ornithine. It was positive slightly for the sulfur, but negative on indole. And all the rest of the sugars were negative on that test. We also did a tryptic soy agar test to demonstrate motility and respiration, and the sulfide indole motility test that also indicated motility. If you look, it's kind of blurry, but there is good growth both on the slant of the agar as well as within the agar itself. That grew out from the initial stab of the agar, and this indicates that the nitrogenomic eutropha will respire both aerobically and anaerobically. You can see here's the control of no growth within the sulfide indole tube, and then you can see growth away outward from the initial stab inoculation with the nitrogenomics. So that it would indicate good motility as well as the anaerobic growth on that. Nitrogenomic eutropha does utilize urea. Urea is an ingredient in our sweat, and it will break it down as indicated by a positive result on a rapid urea test. Once we established the metabolism, we went on and tested how the nitrogenomics will react with potentially pathogenic bacteria. We tested Staphylococcus aureus, E. coli, Micrococcus gluteus, and Staphylococcus epidermidis. These are all common, potentially pathogenic bacteria that are found in our environment and on our skin. And how we did this was immediately after inoculating the nutrient agar plate with the bacteria, 1 plate per bacteria, we soaked what's called a sterile Kirby-Bauer disc in a solution of nitrogenomics in a broth, and using a sterile technique, place that Kirby-Bauer disc in the center of a newly inoculated lawn of potentially pathogenic bacteria, and left it to incubate. This one we incubated in the incubator at 37 degrees centigrade, and the findings are shown here, where we have the disc saturated with nitrogenomic eutropha on the lawn of bacteria. You can see the interaction between the bacteria and the nitrogenomics, with the nitrogenomics inhibiting growth. The pigmentation that you see is the actual nitrogenomic interacting with the pathogenic bacteria and preventing the actual growth. And that growth, you can see, is prevented because this is, on the outside is the normal growth of the bacteria being tested, in this case Staphylococcus epidermidis, and then how it interacts in the nitrogenomic is growing out into the Staphylococcus, preventing it from taking over in that area. In addition, we started to see, okay, how does the nitrogenomics inhibition of the pathogenic bacteria compare to how other antiseptics would work? We have bleach, we have 70% isopropyl alcohol and control. And the top row is the nitrogenomics on the disc. And then we have, let's see here, this would be E. coli, Micrococcus gluteus, Staphylococcus aureus, and Staphylococcus epidermidis. And you can see that the nitrogenomics will interact or inhibit the growth of the pathogenic bacteria in much the same way as bleach. 70% alcohol does not do nearly as good of a job of inhibiting the growth of the pathogenic bacteria. Once we established that the nitrogenomics does inhibit the growth of 4 specific types of pathogenic bacteria, we wanted to see how the soaps and the nitrogenomics interacted with each other. And we tested 10 different soaps: soft soap, liquid dial, Dove, Yes Tomatoes, charcoal black sugar, chia sugar scrub, exotically creamy coconut, Yes to carrot, and then 2 bar soap Clearly Natural Pure Glycerin, and Yardley London Oatmeal and Almond Bar Soap. We determined that the procedure was taken from a lady named Jen, who had done an experiment on her own with antibacterial soaps and how they would work. And I found this online, and so I modified it in order to accommodate our experiment. And what I did was, fresh lawns were made of nitrogenomic eutropha using healthy colonies from a nutrient agar plate that was made into a fresh lawn. In the center of each of those fresh lawns was a Kirby barrel disc that had been saturated in one of the soaps to be tested. These were then incubated at 21 degrees centigrade, room temperature in a dark environment, and they were examined at 72 hours and 120 hours with the observations recorded. For the bar soaps, I had to do it a little bit differently. I used a sterile scalpel and took off a sliver of approximately 17 millimeters by 10 millimeters and placed that in the center of a newly inoculated nitrogenomic eutropha lawn. And again, observed and observations taken at 72 and 120 hours. We have pictures here of 72 hours. You can see on the more natural soaps, such as right here, we have growth clear up to the Kirby barrel disc. On the Dove and Liquid Dial, there is some type of inhibition going on, interaction going on right around that Kirby barrel disc, where it's not really allowing the nitrogenomics to grow. With the soft soap, there was very little inhibition around that. You can see the healthy growth, mucoid growth on the outside edges of the plate. The sugar had quite a bit of inhibition right around the Kirby barrel, but then a lot of growth on the outside. And then you can see some of the natural soaps, the Yes brand soaps. There's growth right up to that Kirby barrel disc, which shows that the soap does not inhibit the nitrogenomics at all. I have a chart here that shows the zone of inhibitions for the different soaps. Again, Dove did not show a whole lot of inhibition. Actually, none was measured with the nitrogenomics. The same with the Zesty tomatoes, and then the Zesty carrots. The one that seemed to inhibit the nitrogenomics from growing naturally the most was the soft soaps, milk and honey. And then there was some with the pure glycerin, the oatmeal, sugar scrub, and then the dial also showed quite a bit of inhibition. At 120 hours, where there was a zone of inhibition where it did not allow the nitrogenomics to grow, we start noticing some regrowth. So this would indicate that after a certain amount of time, the soap is going to lose its ability to inhibit the nitrogenomic, and the nitrogenomic is going to start growing back in. One other thing I want to notice is, like on the Yes with carrots, Yes with tomatoes, and the oatmeal, we have a heavier growth of nitrogenomics right up against the soap. In fact, on the oatmeal one, I actually noticed nitrogenomics growing on the sliver of soap. Here's the chart to show regrowth, changes of pigmentation that were noted between the 72-hour observation and 120-hour observations. The conclusions that I have come to are, again, the nitrogenomic eutropha is a highly motile, gram-negative bacteria. It reduces nitrate, utilizes urea and citrate. Its preferred environment growth is a pH of 6, a temperature of 21 degrees centigrade, and it likes a neutral to slightly saline environment. It produces chromophores when stressed. There's a change of pH or a change in temperature, and also as the bacteria ages. It is able to adequately inhibit the growth of the potentially pathogenic bacteria that were tested, and does so as efficiently, if not more so, than bleach, and definitely better than the 70% isopropyl alcohol. The more natural the ingredients in the soap, the less likely it is to inhibit the growth of the Nitrogenomics eutropha. And generally, it was discovered that approximately 48 hours was needed for the Nitrogenomics to regrow once it has been exposed to soap. Nitrogenomics eutropha has been demonstrated by the experimentation to be considered beneficial to the human body by helping prevent potentially infectious bacteria from colonizing the skin and potentially entering the body. I believe that further experimentation can possibly lead to numerous benefits to humans, with maybe possibly discovering additional potentially pathogenic bacteria that the Nitrogenomics may inhibit, and that if we would reintroduce it to the normal flora of our skin, it would increase the benefits to humans, possibly prevent infections in those working in agriculture, healthcare, or those who are immunocompromised. And the reintroduction of the Nitrogenomics could possibly be done by a spray or a cream. Thank you very much.